Hi students, I am Bukar and this is the second part of our lecture of microbiology. We are going to discuss the physical factor that limits the growth of microorganisms. And previously we discussed the heat as well as the ultraviolet rays which limit the growth of microorganism. Now we are going to discuss the process of filtration, which as we now previously it is one of the most important and rapidly used process for the for the pasteurization as well as sterilization of the materials, etc. This method is mainly used for the those uh, material substances which are heat resistant. As is now, that's most of the substances are in use in lab. For example, different types of chemicals, different types of synthetic media as are the heat resistance. If we autoclave them or use the process of the boiling, etc., then they have caused different then they have ability to denature their original nature and have ability to disturb their function. That's why we use the process of filtration. In the process of filtration, we pass the microbes from a filter uh, which have a appropriate pore size, and whenever microbes pass from this filter, the our media can have ability to uh, move from these pores, but microbes does not move from such pores, and they is in such way where these materials limits their growth. This type of methods mainly used for the sterilization of vaccines, serum, or any the body samples, and similarly the antibiotics. As we know, the vaccines and antibiotics have their specific chemistry, and whenever we increasing their or decreasing their temperature from a specific level, their nature will be disturbed. It is also this method will also use for the purification of laminar flow. What's mean by laminar flow is a chamber in which we are working during our experiment of microbiology. Laminar flow, we want to limit the growth of microbes, and we want to stop the interest of microbes in laminar flow to for the accuracy of our result. In this process we use we use fertilization for the minimizing growth of microbes. Next one is the radiations. Radiations are the powerful and effective and modern tools used for the sterilization of microbes. Radiation on the basis of their nature are of two types. That are the ionizing radiations and non-ionizing radiations. The main difference between these two types of radiation is that the ionizing radiations have ability to penetrate deeply inside the object, while the non-ionizing radiation does not have ability to penetrate inside the substance. For example, the petri dishes. Whenever we are working in labs, we put the petri dishes on one by one, and the non-ionizing radiations does not ability to penetrate in each and every dish and kill the microbes. Similarly, in the case of test tube and other equipment also. The other characteristics of the ionizing radiation is that they have other rapid actions, uh, and the second and most important thing is that these radiations kill the microbes but does not increase the temperature of the media, temperature of the equipments. That's why they does not have any negative impact on their nature. And if we discuss about the how the mode of action of ionizing radiations, these radiations are mainly targeting the nucleic acid and supports. I mean they disturb the process of central dogma and stop the synthesis of different proteins, and as the result of which the death of microbes will be occur. Examples of such radiations are the X-rays, gamma rays, and the cosmic rays, etc. Second one is the non-ionizing radiations. These radiation does not have ability to penetrate, as we previously discussed. We are not able to. These radiations does not have ability to clean up the glasses or other equipments deeply. That's why, due to their non-penetration into the object, they are mainly used for the sterilization of surface of the materials, and mainly such types of radiations are used in operation theaters for the sterilization. And we know that operation theaters are the places where a large number of microbes are present. That's why, to limit their growth, we use such types of radiations. But the main thing is that these radiations are lethal in nature. They have ability to to mutate the DNA of human beings and cause different types of mutation. That may be negative mutations and result different types of carcinogenic infections as well. These types of radiations are the ultraviolet radiations. That's our today's lecture, and in our next lecture we continue this process and we discuss the chemical agents which are responsible for which help to control the growth of microbes. I hope that you enjoy it and get many more. Okay, thanks. Move toward next video. Okay.